Well, hello and welcome to the Wound Healing Society Fireside Chat. I'm Kyle Quinn, co-chair of the Wound Healing Society Communications Committee, and with me is Dr. Mitch Sanders, the past chair of the committee.
Thank you, Kyle. We are pleased to have both Dr. Angela Gibson from the Department of Surgery, University of Wisconsin-Madison, as well as Dr. Lindsay Kalan, who's an assistant professor in medical microbiology and immunology. And I wanted to mention that all of us are somehow Badger fans. Kyle went there, what, for undergrad?
Yep.
My dad went undergrad and MD U Wisconsin before he went to MGH and did thoracic surgery. So it's a very, very small world, which is just wonderful. Why don't we, Kyle, do you want to ask the first question? We'll kind of kick it off.
Sure. And before we get into the this particular paper, maybe if you could both tell me a little bit about your background and how you came together to start working on this project.
Lindsay, do you want to go first?
Sure, I can go first. So I'm a microbiologist by training. Actually, my PhD is in biochemistry, but studying it in microbes. And after my PhD, I started working actually in industry for a wound care company trying to develop antimicrobial wound dressings, which is when I really got interested in trying to understand biofilm and polymicrobial infection and chronic wounds. And I was so intrigued and realized there was a huge gap in the field and our understanding of how we can apply new therapeutics to some of these sort of chronic non-healing wounds or low-key infections that I decided to come back to academia and train at the University of Pennsylvania in the Department of Dermatology with Dr. Elizabeth Grice. And from there, after I did a 2-year postdoc studying the microbiome of diabetic foot ulcers, I moved to my assistant professor position here at the University of Wisconsin, which has just been wonderful because there's such a large, diverse, interdisciplinary group of individuals working on all aspects of wound care and wound healing, which is how I met Angela. And we've been collaborating, I think, since 2018 when I started, and it's been such a great collaboration for us to be able to bring the basic microbiology science side, the way we're thinking about it to our work, and Angela to bring the more clinical translational aspect to the collaborative research. And I'll pass it off to you to talk a bit about your background.
Thank you, Lindsay. And thank you, Mitch and Kyle, for having us on. This is a great opportunity for us. So I'm Angela Gibson. I'm assistant professor at the University of Wisconsin, and I started my career actually as a nurse on the burn unit at the University of Wisconsin over 20 some years ago. I was very interested in science and always asked why about things, and decided I needed to go back onto school because I asked why too much. And so I, at that time, actually got interested in some research going on campus on a skin substitute that ultimately it's now FDA approved, and has been kind of cool to watch that whole journey over the past 20 years. But in that, I got involved in research, and that's how I got interested in research. And my very first paper actually was in Lynn Allen Hoffman's lab, looking at cytotoxicity of the current antimicrobials that I was treating my patients with as a nurse on the skin substitute. And so that was pretty fun to do that translation early on and see that need to have that dialogue between bench science and the clinical science. And so this has been a very cool project because it kind of takes that one step further in utilizing human skin as a more complex model to be able to really test out our theories, our hypotheses. So after I had some experience doing some research, I applied and got into the MD PhD program here at the University of Wisconsin. I'm a lifer here, go Badgers. I did all my training here. And then there just happened to be a burn job here when I was finished with all my training. And so I took that position and developed a lab here, and have absolutely loved the collaboration that I've been able to do with Lindsay. It's just been so much fun. We both we work very well together. It's just a really exciting time, and clearly she's got a lot of expertise that I have no clue about, and so I very, very much value her input.
So maybe that's where it's kind of a great place to start. But could we talk a little bit about the wound excisional model from the human skin as well as the xenograft model in the mouse? Some of the findings that you, kind of the design of how you did it, and then, kind of what concentrations and which antimicrobials or antibiotics did you use for the study?
Yeah, and I'll start out and talk about the cytotoxicity side of things, and then maybe Lindsay, if you want to talk about the antimicrobial part, because we kind of utilized our teams to work on those together, but each other's expertise. So the ex vivo model is a published model. Multiple labs have used it, where you're taking discarded human skin from, say, plastic surgery cases, and then putting them into culture at the air-liquid interface. So you're feeding them from underneath onto a raised platform to allow feeding over multiple days. And so we would create a wound, excisional wound, partial thickness into the dermis inside basically a donut of human skin. We started this out as just seeing if these tissues were cytotoxic to various topicals that we use clinically. And this actually was born from a clinical question. There are some different practices here at the University of Wisconsin in the hospital here about whether we use just saline or whether we should use soap, different soaps or CHG. And, CHG is a pretty ubiquitous cleanser that's used. And some people feel that it's very cytotoxic, but I was kind of concerned that the data was all in monolayer culture or skin substitutes that weren't as robust as human skin. And so we tested, the initial tests were povidone iodine, full concentration for that, Dial and Dove soap, and we diluted that 1-to-1 with sterile water. And then we used saline and then 2% CHG. And those were initially tested inside the excisional wound, and then for 30 minutes, and then irrigated out, and then grown for another 24 hours in culture to see if there was some longer effect of that topical agent. And then for the xenograft model, that we graft a athymic mouse, so immunocompromised mouse with human skin, same skin that we were using in the ex vivo model. And we graft that to an excisional wound that we create on the bilateral flanks of the mouse, and leave that to revascularize. It takes about 8 to 12 weeks, depending on how thick the skin tissue is, to fully neovascularize. It actually goes through an ischemia reperfusion process because of that ischemia that upper layers of the tissue see when we first remove it from its blood supply. And so we need that skin to recover to kind of reset to be more of a normal skin tissue before we can start doing tests on it. So for this specific study, we let them go for 8 weeks. And then we created a excisional wound in that tissue, and then treated those in our ex vivo model. We actually treated it for 2 minutes. It was just CHG versus saline, and we treated it for 2 minutes and then irrigated it 3 times to simulate what we do clinically. And we did that daily for 14 days. And so we were able to see over time grossly what happened to the wound, and then at that 14 days, take the tissue for histology.
Lindsay, did you want to add anything to that in terms of, do you want to talk more about the experimental, the different types of experiments that you did with the study?
Sure. Well, Dr. Gibson just gave a wonderful overview of the model. And so what we brought to it was, asking the question, what's going to happen, under a scenario where you have a wound that perhaps isn't chronically infected or colonized, but becomes contaminated through the process of either the injury or, clinical preparation. So, we approached it saying, can we somehow mimic a contamination model in this, where you may have very, very low levels of bacteria contaminating the surface of that wound? Is using these cleansing agents sufficient to keep that under control and keep the wound clean? So we approached this first using Pseudomonas aeruginosa, because this is a very common wound pathogen. We know that it grows very robust biofilm. And others have used this model to look at infection with Pseudomonas aeruginosa. However, typically they're inoculated with very high levels of the bacteria. So, anywhere from 10 to the 6th to 10 to the 8th. So we started off by saying, what's the minimum number, of bacterial cells required to actually establish colonization of this tissue? And we were able to go down to 10 to the 1. So that's roughly 100 cells of bacteria per wound, which is a very, very small amount. It's right at our limit of detection of even being able to recover it when we do this. And what we found was that, we could inoculate these excisional wounds in the ex vivo skin with just 100 cells and see really robust growth, that by 48 hours after that initial inoculation, we were seeing levels of 10 to the 8, and signatures of biofilm being formed. And so this was really exciting for us that we could do this, because then we could start to look at questions that are based on what happens at that initial contamination stage, and can we really evaluate really the purpose of some of these cleansers, which is just to clean, what's already considered a somewhat clean wound.
So, I always presume that there is no clean wound. I always presume that there's a reason why people do sharp debridement, so that they can get deeper into the biofilm and really remove it. The other kind of question that I had is, I know that the products that you have were kind of off the shelf, but I was kind of surprised to see, 2% CHG. That's 1,000 times higher than the MIC, right? And so I'm not terribly surprised that it's a little cytotoxic. I know Iricept, you've probably seen the bottles in the clinic have 0.05% CHG, and they actually have some really good data on that small concentration of CHG. So I was just a little bit surprised of the product that you chose for the CHG.
So I'll tell you what we do clinically is we pour CHG, for like prepping our surgical cases, we pour straight 2% CHG, and that's what we're using on the wound, which is why I felt that it was necessary to actually do what we do clinically, and why I do think it's really important to test things as we use them. And our burn patients in our unit, we're, diluting it a little bit with water as just to get more substance to it, but we're still using it pretty concentrated, and hopefully not anymore.
We're doing an animal study in a couple of weeks, and we're using the 0.05%, but we're also mixing combination of antibiotics and antimicrobials as well, and looking at the kind of synergistic effects of the different antimicrobials. Hold on, Lindsay, do you want to talk a little bit more about the studies that you did in terms of the metabolic assays and the SEM and kind of the findings of the study?
Sure. So I'll talk a bit about the SEMs, and then I'll defer back to Angela about the metabolic assays. They performed all of those, but one of the questions we had is, typically when we're doing some of these models and studies, we can grow up a biofilm, we can then treat with all of our different antimicrobials. And usually we'll do quantitative cultures to look at what we can recover. But the nice thing about this model, and what we were really interested in, is actually being able to visualize what is happening in the tissue. Where are the microbes colonizing? Are they in a biofilm? So we were really able to take advantage of doing multiple different approaches to somewhat quantify both, qualitatively and quantitatively the microbial burden. So we could do CFU counts. We knew that after treating with these different antimicrobials, we're still able to recover somewhat significant colonies of Pseudomonas aeruginosa, and then later we also tested Staphylococcus aureus. However, what was really interesting is then when we took those same wounds and we looked at them by scanning electron microscopy and then later confocal scanning microscopy, we saw really different things. And so with the SEMs, what we saw is after we treated with CHG, we could not see any viable bacteria or visible bacteria on the surface compared to wounds that were just washed with saline, for example. You saw really dense, thick mats of bacteria and biofilm growing. This was surprising to us because in those wounds that we saw nothing on the surface, we could not see any bacterial cells. We were still recovering about 10 to the 5 10 to the 6 bioburden from those wounds. And so we use the confocal imaging because it allows us to actually go deeper. And what we found was that there were these reservoirs, or, we could see clusters of cells deeper within the tissue that are just not detectable by SEM. And so that suggested to us that you're sort of just doing a surface treatment. It's maybe washing out everything on the top. But these microbes, especially Pseudomonas, this has been reported in literature multiple times, tends to sort of bury deeper into the tissue. And then that can be this reservoir to allow it to then come in and recolonize the wound at a later time point. And that's exactly what we saw in our studies. We saw an initial knockdown of that bioburden when we applied the antimicrobials, but then even subsequent serial treatments of that was unable to really control growth or fully, ever knock it back down, because we just have this reservoir. With the exception of Staph aureus, we did see if you treat and wash early on, especially with the CHG, you could again knock back the bioburden of Staph aureus. And again, this I think is directly related to the MIC, where the MIC is much lower for Staph aureus than Pseudomonas. And again, Staph likes to be at the surface of the wound versus sort of burying deeper into the maybe more anoxic parts of the tissue.
So just a quick follow-up, with confocal microscopy, you're only getting a couple hundred microns deep. How deep were you seeing some colonization with the confocal? Like, was it all the way through your Z-stacks and possibly even deeper, or?
Yeah, that's a good question. We were seeing them pretty deep as far as we could go, and we're pretty good at being able to image with the equipment that we have here, sort of the full area, top of the wound, and then I think several hundred microns deep within the tissue. And so the clusters that we saw were definitely closer to the dermis and deep within the wound. And again, pockets, it's not, nothing was uniform as well. So you're going to see little pockets here and there of, different sizes. And I think one of the key things that we have found from using this model too is that there is a lot of variability, which is exactly what you would expect to see, in a human population as well.
I was amazed and surprised that you don't have cross-contamination. So whenever we do the pig explant model from Greg Schultz, we find a lot of contamination with just these shuttles, and it's really a headache to get. You have to do 9 replicates, and the data is still scattering all over the place. I love that your data was, some of the things we're doing only 4 replicates, and the data looks great, right? We've switched to, we actually 3D print biofilms now. And we're starting to kind of build that so that when you do a 96-well plate biofilm assay, it's the exact same biofilms we can put in the animal, and hopefully that'll control things better. But I was amazed how clean the wounds were, and starting with just 100 bacteria, not ending up with a bunch of things that just came in the theater. When you're dealing with these animals, sometimes they'll peel off their dressing or what have you. It can be a challenge.
So I was just going to say, I think that's the benefit of the human versus the pig, because the pig has a lot more bioburden on it, I think.
Yeah, and I would just comment, because my lab is very interested in the skin microbiome, and we also, do a lot of research just looking at healthy, intact skin and the beneficial aspects of that. One of the very first things we did was really try to evaluate what the bioburden was and what sort of the background native microbiota was on the tissue that we were getting. And what we have shown reproducibly over and over is that it's almost undetectable. So the tissue we're getting is, it's not sterile, but it's for, the purposes of what we're doing. There are very low background levels. We cannot recover either by plating or using, qPCR just to even look for the presence of bacterial DNA with some of this tissue. So that makes it ideal when we want to manipulate it and add different combinations of microbes.
Yeah, we're so used to getting cross-contamination in our porcine studies. We just presume that they have something else, even though we're putting in Pseudomonas or staph or what have you, there's always something else that shows up on the plates. Can we talk a little bit about the metabolic assays next?
Sure. And similar to the way Lindsay was explaining, using different ways to see the same thing, we used an MTT assay as a colorimetric change assay to look for overall viability of the tissue, and then the lactate dehydrogenase staining to look for structural viability within the tissue. And I think that was helpful. The MTT assay is a very fast assay for us to do, and we could, we defined out when we did the initial multiple antiseptics, we were able to see that dial and CHG were the most cytotoxic. What was interesting when we looked at that under LDH staining was that the CHG really affected even deeper, both horizontally and vertically into the tissue. So there was a diffusion of that toxicity that was seen that then persisted as we allowed those tissues to grow over 14 days. And I think that was what was pretty profound and what was helpful about doing a histologic evaluation of viability.
Did you ever take those? I know you did biopsies and got the bacteria out. Did you ever see if they were just now resistant to CHG? Or do you think they were never getting the CHG because it was too deep in the tissue?
We didn't actually. We did do MIC testing on our original stock, not on the cells that were recovered. Although that is something we're interested in, and there are students in the lab asking that question, if after exposure, repeated exposure to CHG, do we see any changes in their overall resistance patterns?
So just a quick follow-up related to the metabolic changes, with the MTT and the LDH measurements. How much do you think that was completely driven by differences in the number of viable cells, or do you think you could also tease out whether there are actual changes in the metabolism of the viable cells in response to the treatment?
Yeah, I don't think I know the answer to that question. I don't think we know.
Kyle, can you share the work that you do on imaging? It's so cool.
Yeah, I mean, this is sort of motivated by the fact that I'm interested in changes in cell metabolism during the wound healing process. And, if you're familiar with what like Melissa Scala does at UW, so I'm using the same technology, looking at autofluorescence changes, but using that to understand differences in how wounds heal. And so, yeah, we're picking up NADH and FAD autofluorescence and finding some differences at the edge of the wound in the keratinocytes. And a lot of those changes really track well with what, say, like Melissa sees with cancers that are, very aggressive versus metastatic and more migratory. So there's a lot of nice, pleasing parallels between the findings that we see in the epithelium of a wound and the dynamic changes in metabolism. I mean, they're really on the order of what you'd see going from normal epithelium to aggressive cancer. It's obviously a well-controlled process most of the time. So yeah, that was sort of driving my question related to the MTT assay, which is you're gonna collect. It's a kind of cumulative measure of everything that's going on in terms of metabolism and the number of cells. Because it looked like there was a couple differences, or some trends, I think, with one of the soap solutions as well, right?
And I think it's getting the whole tissue too, not just the area of wound, which I think is challenging.
Right.
Now these were GFP constructs, right? But they weren't wound isolates. They were just lab isolates that were expressing GFP, so you could just monitor them. We have 250 wound isolates. I've been collecting them for like 30 years. We actually use the moleculite quite a bit. It's a challenge. You really have to get used to seeing the fluorescence, the bacterial fluorescence with the moleculite. It's a little bit of an art form. And usually I have 3 people read the wounds, clinical wounds blinded, and then we compare notes, and then we always have discuss at least 3 or 4 wounds that we have an issue with. Did you use 2 different you didn't do Pseudomonas and staph together, right? So it wasn't 2 different GFPs? No. Okay.
No, yeah, they were just mono infections for this study.
And because staff takes longer to kind of make a nice biofilm, that's why you did the 48 hours?
Yeah, the staff look took a little bit longer to establish. We also found because it is more susceptible to CHG, we did do a washout period because the tissue is surgically prepped with CHG. So that was another, I think, really important finding actually just for anyone who wants to use this model in their work. We did find that we had to have sort of a washout period to make sure anything that the patient was treated with prior sort of washed out of the tissue, and then we could start our manipulations and colonizations.
Yeah, if I ever have surgery in the next year, I'm going to make sure it's a lower concentration of CHG or something. So what's next on the horizon for this project? Where do you see, are you thinking about looking at multi-species communities? I love looking at, at least 3 different, these wounds are usually polymicrobial anyway. So I love looking at anaerobes, and I love looking at, how the Pseudomonas interact with the staph and the complex bacterial community that we don't get to see in the Petri dish.
So, I mean, we're really interested in polymicrobial communities, and so that's something we're trying to establish. And although clinically in a human patient, you might find these really diverse communities in the wounds, it's much harder to mimic in the lab because we are still sort of putting them into a different environment, and we don't really understand all of the factors guiding this mechanistically. Sorry, did I lose you?
No, I'm
Okay.
still
But we're trying to build complexity. So right now we have a 3-member community we're working with, but I have several PhD students who are thinking about this and thinking about ways to design these microbial communities that would reflect what we're seeing in wounds in the clinic, and thinking about ways to then put that into this model, whether it be just to look at the basic, again, mechanisms that are sort of driving community assembly, or what happens when you have a polymicrobial community and its resistance or tolerance to some of these antimicrobials and antiseptics compared to the mono infection. And I think I'll default now to Angela, but I think the other question is, how does that impact tissue viability and other immune processes when you have different types of polymicrobial infection versus single, and different treatments for those?
Yeah, I mean, I think it's going to be a hard sell for me to go to the hospital and say, okay, everyone's got to get rid of CHG because of what we found here, right? And so I think it's an interesting finding. I think, we have to find something that we would have to use in place of that, and could it have the same or better efficacy, as well as less cytotoxicity? So that would be interesting to follow up. And I also think, is it different between an excisional wound versus a burn wound versus a chronic wound? All of these different things, or intact skin before it's been wounded. Are there differences? Is there something about the intact skin that actually prevents it from becoming a problem? Is the diffusion into the wound itself what the problem is? So is it okay for us to use this as a preoperative scrub? So those are, and then the other thing I'm interested in is, us all using these pig models, and how really translatable that is. Everyone says pigs are the, great model for human skin, but there are quite a few differences between pigs and humans. And obviously we can't experiment on our patients. So we have to have some surrogate, but just recognizing the limitations of those models.
Some of the best porcine burn studies are done at Wisconsin. Are you involved in those as well, or are you just other colleagues at the university?
I don't know of a whole lot of burn stuff going on in pigs here.
I just reviewed a paper last year.
Okay.
Really good burn model from Wisconsin. That's the reason why I know, and we're looking at kind of replicating it here.
Yeah.
Paper. It's probably somebody right down the hall from you.
Probably. It's such a big university.
So I just have to say, this was just a wonderful chance to meet you both. Wonderful paper, and we're just delighted to have you hosted on the Fireside Chat. And hopefully you'll get to come to the meeting. Do you guys both go to SAWC WHS in the spring? Great.
Yeah.
Well, I host all the social events, so please come to our. We do the innovation award. We do what we call Wound Shark, where we give out an innovation award once a year, and we somehow also sponsor the social event for the Wound Healing Society. So hopefully you can make it. And Kyle, are you going to innovations this year or no?
Yeah, I'm thinking about it. I think the abstracts are due this week. So I'll know by the end of the week.
I don't have a boat yet, but I'm going fishing again. So if either of you want to go on a fishing trip, if you go to Innovations, I usually take clients fishing. It's actually quite fun. Kyle came last year. We caught a bunch of, what was it? Was it snapper or it was
I'm blanking on what it was, but caught a decent amount.
Yeah, then we brought him back to the restaurant and they cooked him for dinner. A whole bunch of friends came for dinner. It was pretty exciting. So anyway, anyone going to the Boston meeting coming up in 2 weeks at Beth Israel for wound care? So I'm doing that, and then I'm going to Innovations, and then I'm giving it to Germany week in Berlin for another wound care conference. It seems like there's one almost every week now. I don't know how I'm going to keep up. But anyway, thank you kindly. Wonderful to host you on the Fireside Chat, and we'll keep following your careers. And if we can ever collaborate, we'd be delighted.
Well, thank you so much for the invitation. It was great chatting.
Yeah, great to meet you guys all.
Take care.
Thank you.
Bye-bye.
Bye.